Hi, I'm Bill Rapisi, Executive Director of Lymphatic Education and Research Network, and I just want to take this opportunity to thank all of you for joining us today for our symposium series. I also want to take this opportunity to thank all of you who have become members of LE&RN and help support this series so that we can continue bringing this kind of groundbreaking information to you in your homes and offices. I would also suggest for all of you who have not become members of LE&RN, $5 a month, you can become a member of LE&RN and help support this programming and the research that we do. Thanks very much for joining us, and I look forward to having you become a member.
So hi everyone, I'm Echoe Bouta. I'm a postdoc research fellow here at Mass General, and I want to thank LE&RN for the opportunity to give this symposium today on an overview of the current state of lymphatic research. So this talk is by no means all-inclusive. I've really wanted to highlight some of the diseases that aren't typically thought of as being lymphatic-specific disorders that have recently been found to potentially have a lymphatic component. Okay, so an outline of my talk, I'll first go through an overview of the lymphatic system as a whole, and then I want to just briefly go through some of the current diseases that there's active lymphatic research going on in. And then I'll finish up with conclusions. And so I first wanted to point out that lymphatic research is really increasing exponentially. And you can see this exponential increase started around 1990, and that is by no means a coincidence. So it was around that time that the first lymphatic-specific cell markers were found, and that really gave researchers the tools they need to perform research on lymphatic vessels. And so the reason why I bring this up is because a lot of other fields found their cell-specific markers much earlier, such as in the 1960s. And so the lymphatic field is still very much in its infancy, and I think we're still learning just how important the lymphatic system is and all of the diseases it could be playing a role in. So as many of you know, there's three major functions of the lymphatic system. The first is that it absorbs and transports fat from the digestive system. The second is that it regulates interstitial fluid balance. So here you can see this capillary down here where you see fluid and macromolecules travel from the capillary through the interstitial space and into these lymphatic capillaries that are also referred to as initial lymphatics. And so you can imagine if this lymphatic capillary wasn't here, or if it wasn't functional, you would get this buildup of fluid and macromolecules in the interstitial space. The third role is that the lymphatic system is really important for immune surveillance. And so you have lymphatic vessels almost everywhere in your body, and your immune cells use this as a highway to check to see if there's any pathogens that might be a threat. And then they use this transport system to teach other cells about what they should maybe be targeting. And you can see this immune cell down here that's entering the initial lymphatic. And so the lymphatic system somewhat starts in the initial lymphatics, or lymphatic capillaries. And these are simple vessels that are simply a layer of endothelial cells. And these have specialized junctions that form these primary valves that allow fluid to enter. And so these are essentially flap-like junctions, so if the pressure in the interstitial space is higher, it'll drive fluid into these initial lymphatics. So once fluid leaves the interstitial space and enters the initial lymphatics, it's what we refer to as lymph. These initial lymphatics drain to the collecting lymphatics. And here you can see these vessels have muscle cells that allow them to contract. So in mammals, there is no lymphatic heart, unlike the cardiovascular system. And so this means that these vessels themselves have to contract to move fluid against the force of gravity. So again, here you see this normal lymphatic contraction that's occurring. And in inflammatory conditions, you can see that the vessels are not contracting as well. And so an active area of research is understanding how these lymphatic vessels respond to inflammatory conditions and if their response can be modulated to help clear inflammation faster. These collecting lymphatics then drain to lymph nodes. And these lymph nodes are specialized organs that are really the powerhouses of the immune response. And so you can see that they're chock full of immune cells, such as T and B cells. And so the way the immune surveillance of the lymphatic system works is you'll say you have an infection in your toe. an immune cell will capture part of that bacteria and will enter the initial lymphatics, and then will transport it all the way down the collecting lymphatics to the draining lymph node. And in this draining lymph node, it'll educate the other cells there about what the threat is and help teach them how to combat this threat. And this is also where these cells mature and form a memory response. And so I just wanted to briefly mention lymphangiogenesis, as it's something that comes up in the coming slides pretty often. And so I'm going to boil it down to its most basic and classic pathway, and that is a VEGF-C pathway. So here you have VEGF-C, which is a protein, and this binds to a receptor called VEGFR3, and this triggers lymphangiogenesis of the lymphatic endothelial cells. And so lymphangiogenesis occurs often in the body in cases of inflammation or injury. And so as an example, you can see here they've removed the lymph nodes, and you can see this one lymphatic vessel. And then if they give VEGF-C, you can see all of these lymphatic endothelial cells have formed somewhat of a sheet in response to that. And then over time, you can see they mature and become more functional. So next I want to start with the current diseases of lymphatic research. And so I feel compelled to at least mention lymphedema, as it's the cardinal manifestation of lymphatic dysfunction. However, LE&RN has had many symposiums with experts of the field, so I'm by no means going to go into depth on this topic. And so lymphedema comes in two flavors. The first is primary, which is usually due to a genetic mutation that causes the lymphatics not to form properly, and you get this somewhat widespread swelling. The other flavor is secondary, and this happens in response to an injury or an infection. And so in the Western world, we typically think of it as occurring after treatment of cancer, where they remove the lymph nodes. And that removal of lymph nodes causes an obstruction of the lymphatic system and means that the lymphatic system can't drain the fluid of the affected limb. And so you can see in this patient, one leg is affected due to the fact that the lymph nodes were only removed on that side of the body. And as I'm talking about current research, I wanted to mention that recently a phase 1 clinical trial has been started in Europe using a VEGF-C gene therapy to treat breast cancer-associated lymphedema. So next I want to move on to cancer. So I think cancer is one of the classically thought of diseases whenever you think of diseases with the lymphatic component. So I'm not going to go into too much detail with this, but I just wanted to mention that there is an active area of research looking at how these lymph nodes respond to cancer. And so before the cancer cells even get to the lymph node, in the pre-metastatic lymph node, you can see that the lymph nodes are enlarged, there's increased lymph flow, there's lymphangiogenesis. And so the thought is that these tumor cells sort of prep the lymph node for their arrival. In the metastatic state, you can see these tumors arrive and cause further changes to the lymph node. So another area of research is understanding why these lymph node tumor cells are not being fought against, despite the fact that they're surrounded by the body's immune response. So next, moving on to Alzheimer's, I wanted to take a few minutes and introduce a recent discovery that is the glymphatic system. So although there are some older publications suggesting the existence of a brain lymphatic system, the current dogma in the field was that the brain did not have a lymphatic system, which never really made sense because it's a large organ that's very metabolically active. And so it was unclear how the brain was clearing all of its solutes and interstitial fluid. And so recently a group discovered what they refer to as the glymphatic system, which gets its name because it's similar to the lymphatic system and is based off glial cells. And so what they found was that fluid can move paravascularly, so around the vessels, and can travel through the brain interstitium through these pathways, and then eventually leave next to the venous return. And they found that, importantly, this glymphatic system is dependent on a protein called aquaporin 4. And so I bring this up because in terms of disease studies, this brought up an important question about Alzheimer's disease. So Alzheimer's disease, one of the prevailing theories as to the cause of it is that there's a buildup of a protein called amyloid beta. And so whenever the glymphatic system was discovered, people started to question if this glymphatic system could drain some of that amyloid beta. So what this group did is they injected 2 types of amyloid beta. The first was a radioactive amyloid beta, and with this, they could then track the radioactivity of the brain and see how fast that amyloid beta was leaving. And what they found was that whenever they looked at mice that didn't have aquaporin 4, that protein that was very important for glymphatic system function, that there was higher levels of amyloid beta in the brain compared to in the wild-type mice. The other type of amyloid beta they injected was this fluorescently labeled amyloid beta. And they did this so that they could look at how it was moving through the brain. And you can see these very defined tracks at which the amyloid beta is moving through the brain, suggesting that it's not just freely diffusing and is actually following this glymphatic pathway. And so the hope is that if we can trigger the glymphatic system to be more active, then maybe we can help clear some of this harmful protein and prevent the onset of Alzheimer's. I also wanted to take a minute to introduce the fact that although, as I mentioned, the dogma that the brain does not have lymphatic vessels, it was recently found that they do have lymphatic vessels in the dura, so the outside of the brain. And you can see that they follow a lot of the blood vasculature of the dura, similar to how the lymphatic vessels follow the blood vessels in the periphery. And so in this paper, they also demonstrated that these lymphatics are functional, and if you inject protein into the brain, it will in fact drain through these lymphatics to the cervical lymph nodes. So they are functional. So next I want to move on to the eye. So cornea grafts are usually successful, but occasionally you'll have a high-risk patient where the graft is rejected. And so in that case, we need to come up with new treatment strategies. So in this group's research, they look at both models of high-risk and low-risk graft survivals, and you can see that high-risk models show a much lower % survival of the actual cornea graft itself. And in this case, you can see that high risk shows much more blood and lymphatic vessels present than in the normal low risk that you see here. And so they started to wonder if reducing some of this lymphangiogenesis and some of this blood vessel genesis, the angiogenesis, might be a way to help promote survival of the cornea graft. And so first what they did is they treated the mice with a VEGF trap, and so this blocked both angiogenesis and lymphangiogenesis, and you can see that there is no blood vessels in the cornea. And then you can see that this does significantly improve the survival of these high-risk grafts. Then they asked if just blocking VEGFR3 or lymphangiogenesis could have a similar effect. So here you can see whenever they block lymphangiogenesis by blocking VEGFR3, that there is still blood vessels, but there are no lymphatic vessels. And despite the fact that there's still blood vessels, you can see that there is still a high % survival in these animals, suggesting that lymphatics are very important in this cornea graft rejection. So keeping with the theme of the eye, it was recently found that a canal in the eye called the Schlemm's canal does have some lymphatic markers. So here you see the Schlemm's canal that's staining for PCAM1, which is an endothelial marker. And then you can also see that it stains positive for VEGFR3, meaning that it is in fact lymphatic. So the next thing they did is they treated these mice with VEGF-C to see if they would respond. And you can see that in fact there is massive lymphangiogenesis that occurs during this treatment. They then measured intraocular pressure in these animals. And you can see that in the VEGF-C treated mice there is a much lower intraocular pressure compared to in the control. And so this is important, as in glaucoma, the higher the intraocular pressure, the higher the risk of glaucoma is. So the hope is that if we can reduce intraocular pressure by improving lymphangiogenesis, then maybe we can come up with a novel treatment for glaucoma. So next, moving on to cardiovascular disease. So it's been long known that lymphatic vessels run parallel to blood vessels. However, it's becoming more apparent the extensive crosstalk between the two types of vessels. And so one of the surprising things that was found was that there's some dependency on lymphangiogenesis effect on the hypertension. And so here you can see they gave rats either a low salt diet or a high salt diet, and then the mean arterial pressure is higher in the high salt diet, meaning that these rats are hypertensive. They also did histology in the skin, and you can see that those rats also presented with a higher lymph capillary density, meaning that there is lymphangiogenesis that's occurring. So that they then questioned if this lymph capillary density actually is playing a role in the disease. And so they blocked the lymphangiogenesis, again by blocking with the VEGF-R3, VEGF-C pathway. And they see that that does in fact lower the lymph capillary density, and that in those animals that were treated to block lymphangiogenesis, there is a higher mean arterial pressure, suggesting that lymphatics are important for hypertension. And they further illustrated this, as you can see in animals with a very high lymph capillary density, they have a much lower arterial pressure compared to mice with a low lymph capillary density that have a high mean arterial pressure. So in a mouse model of myocardial infarction, where they ligate one of the blood vessels of the heart, you can see this extensive damage that occurs over time. And if you look at the damaged area, and you stain for lymphatic markers, namely podoplanin and LIVE-1, you can see extensive lymphangiogenesis is occurring in this injured area, as we expect, because lymphangiogenesis occurs often during inflammation and injury. So they questioned whether or not these lymphatics are important and whether or not this lymphangiogenesis could be modulated to have a treatment effect. And so in their control animals, you can see that after myocardial infarction, there's very little difference between the diastole and the systole. So the diastole is whenever the heart is filling with blood, and the systole is whenever it's contracting. And the fact that there's very little difference means that the heart is not efficiently removing the blood, which means it'll have a lower ejection fraction, and you want your ejection fraction to be as high as possible. Whenever they treated these animals with VEGF-C, you can see that the systole now is much smaller than the diastole, suggesting that the ejection fraction is higher, which is quantified here. And so the hope is that if we can trigger lymphangiogenesis more extensively in these animals, then maybe we can improve the recovery after a heart attack. So next, moving on to asthma. So there's been a little bit of clinical work done on this arena, where here you can see in controls type 1 and type 2 asthmatics, there is in fact a correlation between the airway radius and the area of lymphatics, suggesting that lymphatics are important for sort of mediating airway function and possibly the edema that occurs in asthmatics. And so in these animals, they also infected with bacteria. In animals, they infected with bacteria as a model of lung inflammation. And you can see with no infection, there's already a lot of blood vessels and lymphatic vessels in the trachea. However, after infection, you see this extensive lymphangiogenesis and angiogenesis that actually persists. And this group has blocked this lymphangiogenesis and see that the edema of the lung actually is worse whenever lymphangiogenesis is blocked. So the next steps are to find a way to take advantage of this lymphatic vasculature to help clear some of the edema and the immune cells that occur during asthma and other lung inflammation. So moving down the body to irritable bowel disease. And so as I mentioned, the lymphatic vessel contraction is often modulated by inflammation. And so this is a great example of how that occurs, where in guinea pigs that have been given TNBS that induces inflammation in the bowel, there is less lymphatic contraction compared to the sham. And what they found is that if they give a drug that targets lymphatic contraction, they can actually restore it, which is quantified here. And so this is one of the first groups that have really gotten to not only the presence of lymphatic vessels, but also the function of them and how well the collecting lymphatics are contracting. And so the next step, I would guess, for this group would be to see if restoring lymphatic contraction can actually reduce some of the side effects of the inflammatory bowel disease by reducing the immune cells and the edema present there. So it's been long known that lymphatic vessels reside within adipose tissue. However, understanding the crosstalk between the two has not been extensively studied. And so my work specifically is on a disease called lymphedema, which is where women get increased adipose tissue on the lower limbs. And this adipose tissue is very stubborn and hard to get rid of, so calorie restriction is largely ineffective. And lymphatics come into play as a lot of these patients show increased fluid retention that worsens as the disease progresses. So here on the left you see a patient with stage 1, while here on the right is a stage 3 patient that is also referred to as lymphedema due to the extensive fluid retention that these patients have. So this suggests that there is a lymphatic dysfunction that maybe can be targeted. A similar disease is called Dercum's disease, where patients get lipomas all over the body. And so there's been some lymphatic imaging done in patients. And so here's a healthy human patient where you can see the dye was injected here and it's filling these lymphatic vessels. And these lymphatic vessels are very linear. You can see occasional packets of dye transporting through, suggesting that they're contracting. And this is in contrast to Dercum's disease, where there's no such pattern of the lymphatic vessels. And also you can see this physician is massaging this lipoma right here that appears to be taking up the dye, suggesting that it might have some sort of a lymphatic component. And so unfortunately this field is still very much in its infancy, but hopefully in the coming years we can make some headway into these diseases. So now going back to mouse research, it's been recently discovered that high-fat diet actually impairs lymphatic contraction. And so here you see a normal chow-fed mouse, meaning they were fed their normal food, and this is a collecting lymphatic vessel. And you can notice the differences between this and a mouse that's been fed a high-fat diet, because the diameter of these vessels are much larger compared to the chow control. So this group also did functional assays of lymphatic contraction, and they found that the high-fat diet mice show a lower lymphatic contraction frequency compared to chow-fat diet. And keeping on that theme, there's one of the best ways to study lymphatics is by genetic tools. And so PROX1 has been identified as a gene that's absolutely necessary for the lymphatic lineage, meaning if this gene is not fully active and working or present, that the mice will have a lymphatic dysfunction. And so they took these mice that had a PROX1 haploinsufficiency, and you can see that these mice weigh more compared to their wild-type litter mates. And if you look at the adipose tissue, you can see the wild-type fat cells are nice and small in comparison to the PROX1 haplotype mice. And so this is really interesting because the only thing about these mice should be the lymphatic insufficiencies. And this group also did demonstrate that there is lymphatic insufficiencies. So here you see a wild-type thoracic duct that's very clear, where in the PROX1 mice there's a lack of this thoracic duct, but there seems to be compensatory mechanisms. So there's definitely some sort of a lymphatic vascular defect that possibly is contributing to this increased adiposity and this obesity that occur in these mice. In this paper, they also took lymph from these mice and showed that it was in fact adipogenic, meaning that it did cause adipocytes to proliferate. So next I want to finish on a disease that's near and dear to my heart, which is inflammatory arthritis, as this is what my Ph.D. was focused on. And so inflammatory arthritis mouse models are essentially models for rheumatoid arthritis. And so in this mouse model, we performed contrast enhancement MRI imaging of it. And you can see on one leg of this animal, there's very little inflammation, and the corresponding lymph node behind the knee is very big and bright, indicating that there's likely good drainage to that lymph node. On the contralateral side, in the same animal, you see this extensive inflammation that's occurring, this synovitis. And the corresponding lymph node is much dimmer, suggesting that there's less flow to that lymph node. And so if you look at the volume of these lymph nodes, you'll notice this very bright lymph node is in fact much larger than this dim lymph node, which is why we refer to these two phases as expanding and collapsed. But what's most important is that during the collapse phase, you see this increase in joint destruction. So we began to wonder if the lymphatic system could be contributing to the lack of drainage of some of the harmful cytokines in the joint, contributing to this joint destruction. So the group did lymphatic imaging, and so this is done by injecting a dye into the foot pad and allowing it to fill in lymphatic vessels leading up to the popliteal lymph node. And you can see in the collapsed mice, so the ones that have a lot of joint inflammation, the lymphatic vessels are very dim and aren't taking up much of the dye. And this is in contrast to the expanding node mice, where you can see that the lymphatic vasculature is very bright. So if you place a region of interest on this vessel, you can actually look at the lymphatic contraction. And so they found that wild-type and expanding lymph node mice have a very similar lymphatic contraction, where mice that had a collapsed lymph node showed no lymphatic contraction. And so the current area of research is trying to find ways to restore this lymphatic contraction, is the hope that restoring the function of those lymphatic vessels will help drain some of those inflammatory cells and cytokines from the joint space, helping to reduce the joint inflammation. So in conclusion, I hope I convinced you that lymphatics are important. They're essential for many critical functions in the body. Research in animal models suggests that they play an active role in several disease pathogenesis. However, not much of this has been translated to clinical trials, so hopefully that's coming in the future. And hopefully this demonstrates that lymphatics should at least be looked at as potential treatment targets due to their extensive role in many of these diseases. And so for acknowledgments, I first wanted to acknowledge the fact that I'm part of what we refer to as Team Lymphatics of the Steel Lab, and we're led by Tim Padera. I also want to acknowledge Ilko Meyer, who's here helping me out today with the technical aspects. And I'd like to thank LE&RN and the Fat Disorder Research Society for funding support. And so with that, I'm happy to answer any questions you may have.
There's a question. What do you think will be the first lymphatic-specific therapy to make it to clinic?
So the question is, what do I think will be the first lymphatic-specific therapy to make it to clinic? So currently, there has been phase 3 trials completed on stopping angiogenesis and lymphangiogenesis in cornea graft transplants. So I think that that'll probably be the first one that'll make it to clinic.
Another question. What do you think are some of the boundaries in understanding lymphatics in a clinical setting?
So the question is, what do I think are some of the boundaries of understanding lymphatics in a clinical setting? And I think some of the major boundaries right now are the fact that it's difficult to actually see and visualize and quantify function of lymphatics in clinic. Some of the techniques are kind of lacking. But the slide where I showed with Dercum's disease, I think is a promising new way to look at lymphatics in clinic, and so hopefully that'll become more commonplace so we can assess the function in different disease cases and understand if these patients truly have a lymphatic dysfunction, and we should be looking at other treatment options.
Another question. You presented a lot of animal research. Is there very much being done on human subjects?
So the question is that I presented a lot of animal research and if there is a lot of research being done on human subjects. And the answer is no, unfortunately, at least that I've found. So as I mentioned, there is the phase 1 clinical trial starting with the breast cancer-related lymphedema. There's the phase 3 trials that were recently completed for cornea grafts. And then other than that, there's been imaging done in several other disease states with the near-infrared imaging, which is the imaging system that I showed with the Dercum's patients. And so that's been really one of the best ways to look at lymphatics in patients, as a biopsy isn't required for that.
Next question. How can one increase lymphangiogenesis?
So increasing lymphangiogenesis, the question is, how can you increase lymphangiogenesis? So lymphangiogenesis occurs pretty well on its own in inflammation, as there's a lot of things that trigger it. However, if you want to increase it, I think VEGF-C is the most commonly used protein to induce lymphangiogenesis. However, it's recently becoming more and more apparent that there are other pathways that are important for that.
LE&RN says thank you from all of us at LE&RN for a fascinating symposium.
Okay, thank you all.